So welcome everyone to the third XLA Life Fireside Chat. I'm Austin Stack. There are no guest speakers today. And today's talk is going to be all about B cells. So on the agenda, we're going to meet the B cell. We're going to go through the steps of B cell development. And then finally, we're going to go through the so what. Why does this matter? Why do XLA patients need to know about B cell development? We don't have B cells, so why does it matter, basically? And I want to start out with a quick disclaimer. This is not going to be your usual webinar. I'm putting my own personal style on this, and it's really been influenced by two major educational resources, starting with McGill University. I had a professor in my intermediate immunology class give us a really great homework, and it was draw a cartoon on your favorite immunological process. So I did it on B cell development, and you can see my rough draft on the screen here. This is really actually B cell activation. And I really enjoyed that, found I learned more than any test, any flashcards. But I wanted to make it a little more cleaner and also something that anyone can understand. And so the second influence was this subreddit, so off of reddit.com, called BioBall, and they make pretty humorous cartoons about immunology. I think it's really cool how they personify the cells. I think it's a great way to remember things. And so this is a great example of a comic that I found on the subreddit from username I am Bat Penguin. So being a white blood cell means you have to protect against all kinds of menaces, viruses, bacteria, and most importantly, whatever the blank these are. So it's shrimp, pollen, and peanuts, because white blood cells often, for some reason, feel the need to mount a response against things like pollen and peanuts, and that's what causes allergies. And we're going to go into why that occurs and how the body usually can prevent these cells from getting past the B cell development stages. So the second disclaimer on that note is that I drew most of the following images. So if you see anything poorly drawn, you can please send me an email. I'm looking for an artist. But I had a lot of fun drawing these. It was quite tedious, but I look forward to doing more. And if this works out well, I'd love to hear all your thoughts and what I can improve on. And then the third disclaimer is a little more serious. None of this is medical advice. I'm not a doctor. I am just a guy with a passion for immunology, and I have XLA, so I wanted to draw about the B cell. And so after those disclaimers, we are going to get into the meat of this fireside chat. And it begins.
You may have to trademark or copyright your cartoon images.
I would, yeah, if it gets a good enough response, may actually end up posting these online. We'll see. So we start out with, what is a B cell? Well, a B cell is a part of the adaptive immune system. So there are two main branches of the immune system, and I think of them like an army, a country. Your body is a nation, and the two main branches are the innate immune system and the adaptive immune system. So the innate immune system is always there. They're like always there, always active. They're frontline soldiers, the first ones on the scene. So it includes things like physical barriers such as your skin, mucosal surfaces, but also many specialized cells like macrophages and neutrophils. And we'll see some of those later. The adaptive immune system is made up of T and B cells. And so B cells are a main branch of the adaptive immune system. And it's best described by the vaccination process. So the adaptive immune system gets shown a picture. You get a vaccine. It's basically a snapshot of a pathogen, and then it will adapt to fight off whatever is shown to them. And it will remember this picture and be ready for the next time it sees that pathogen. And that process can take a full week or two. before the adaptive immune system mounts an effective response against the initial infection. But later on, it will be ready immediately from the moment it sees that picture again. And so B cells use a tool called the B cell receptor, or BCR, and antibody as its weapon of choice against pathogens. And so what is a BCR or antibody? Well, it's this thing on the surface of the B cell. So the B cell receptor is bound to the surface of this B cell here. And then a little humorous joke for the antibody, it's a BCR until it is antibody, and it is now off of the body of the B cell. It gets secreted out. Now there are exceptions. There's membrane-bound antibodies, of course, as well. But I thought I'd give my go at a little joke there, a little nerdy joke. And also, our final point on B cells is that there are several types. So here we have 3 types here. So here we have a pre-B cell with its B cell receptor on the membrane. In the middle here, we have a plasma cell, which is a fully matured B cell, and that just pumps out lots of antibody. And then we have another type over here called a marginal zone B cell. These live in your spleen and pump out a different kind of antibody, one you might not be used to, called IgM. And so it's not limited to these 3 types. There are several more, but I needed to keep it a little more simple rather than drawing out every single type of B cell. And so we move on. And if the immune system is an army, B cells are the artillery of the immune system, and antibodies are the shells being fired. So we have a B cell firing out antibody at this bacteria, which is running away. And these antibodies have a few different functions that they can do to help the immune system. One of them is opsonization. So this is when antibodies bind to a pathogen and kind of slow it down, and then it tags it for elimination. And here we have a macrophage that can now recognize and eat a bacteria that has been opsonized by antibody. There are other functions such as cross-linking. So cross-linking occurs when antibody bound to something is recognized by another immune cell, and this will activate the other immune cell. This is common for mast cells. Many other immune cells will be activated by the presence of antibody. So that's an introduction of B cells and what they are and what they do, what their function is. So how does a B cell become a B cell? We begin with this chart on the right. So this is from a textbook called Janway's. It's immunobiology. It's basically the holy Bible of immunology. You could really teach yourself immunology just from this book. And don't worry about any of these bottom boxes. We are just going to deal with the stem cell through pro B cell, through pre B cell to immature, to finally the mature B cell. And we'll be referencing this chart later. So some of the goals of B cell development are to generate diversity. And what I mean by that is that, well, let's begin with a little thought experiment. So the immune system is there to fight off foreign invaders. Well, how many foreign invaders are there in the world? It's basically everything besides yourself could be a foreign invader. And there are trillions of possible antigens, things we need to fight off. And so we need to create trillions of different B cells, or at least antibodies that can possibly fight off anything we come into contact with. And so there's a single antibody might bind to a single antigen. So we need a different antibody for every antigen. And we do that by generating diversity. And we'll see in a couple slides how that occurs. We need to encourage foreign reactive cells to mature. So that means B cells that react to foreign objects are the ones we want to mature. Conversely, we want to stop autoreactive cells from maturing. And that means we don't want any cells that react to antigens that are ourself. That's how autoimmunities like lupus arise, when B cells activate to antigens of yourself, and then you have B cells producing antibodies that attack your own body. So those are the goals of B cell development. And now we're going to go through each step in this chart here, starting with the stem cell. So the hematopoietic stem cell, we begin in the bone marrow. Now, everyone has stem cells in their bone marrow still. Even as you get older, you will still have these kind of baby naive stem cells growing. And that is because they have a few superpowers, one of them being self-renewal. So they're able to grow and divide and stay stem cells themselves. And the biggest superpower of the stem cell is being multipotent. And we'll get into that in the next slide. But as I said, we are in the bone marrow right now. And as you see in this image, the bone marrow isn't just hard white bone. On the inside, it's very vascularized. There's lots of blood vessels, lots of nooks and crannies for blood cells to live in and interact. And there's also other cells besides the stem cells, such as a type called a stromal cell, and we'll see what role they play in a second. But these stem cells are targets for gene therapy, which is why we activate your stem cells to leave the bone marrow, because we need to get them out. And these stem cells are kind of looking for a job. They're looking for a function. And these stromal cells, aforementioned stromal cells that live in the bone marrow, basically ask the stem cells, hey, what do you want to be when you grow up? And they kind of give them a hint in the form of this protein called IL-7. IL-7 basically pushes this stem cell to want to be a B cell. It will retain it in the bone marrow until it develops into a bigger B cell. And so that's how a stem cell basically gets pushed towards the B cell route. Let me talk about the idea of being multipotent. So multipotent means it's able to take on several different jobs. And that's why looking for a job is in quotes. If we go back to Dr. Rawlings' slide here, we see the hematopoietic stem cell is at the root of all of these final cells, from red blood cells, erythrocytes, platelets, eosinophils, monocytes, macrophages, T cells, B cells. It all begins with the hematopoietic stem cell. It has the ability to take on all of these jobs. So that's why the stromal cells sending out IL-7 to tell these stem cells to become B cells is a major step. And we're gonna really ignore the bottom part of this pathway here because those aren't B cells. And we're also gonna skip through the common lymphoid progenitor and lymphoblast. We're gonna start off in the beginning stages of the B cell. And so the first stage of the B cells are pro-B cells. And a really important and complex process begins here. And that process is called recombination. So just so we know where we are in the bone marrow still. We've just become a pro-B cell. And what recombination is, it's basically, we need a way to mature as a B cell. Right now, we don't really have a function. We don't have a B cell receptor. We don't have antibody yet. Instead, we have this DNA and all of these segments. So this is such a complex process. So as I said before, we need trillions. We need the ability to create trillions of different antibody. And the way we do this is with this little segment of DNA. And this segment of DNA is in every cell pretty much, but B cells have this miraculous ability to cut and alter their own DNA. They can mutate, same with T cells. And this is how we generate diversity. So how does this happen? Well, we get this protein here, starts getting expressed. It's called recombinase. And this moves along and basically starts to cut DNA in different segments. So it starts in J and D, and it can do it in many different combinations. And so let's say we cut at J2, D3, and B2, then we get this shorter segment. And basically it will continue to cut, try different combinations of DNA until we get a B cell receptor on the top of the B cell here. And it will only stop once there's a functioning B cell receptor. It will try many different combinations. So I wish I had the ability to drag each, like J1 here. It could be J1, D9, V5. There are several different segments. And that's how it offers the ability to have really trillions of different antibody types. And another important note, side note that I want to mention, because it's a bit of foreshadowing for our next Fireside Chat, is when we cut this DNA, the DNA that gets cut is released in the form of what are called CRECs, excision circles, basically. And these CRECs can be used as diagnosis tools. So if you don't have these cuts of DNA being released, that means you're not generating different B cells or T cells. So if you don't have these CRECs or excision circles release DNA, then that's an easy way to diagnose things like XLA. You won't have any CRECs in XLA because there's none of this DNA cutting occurring. Or for SCID, you won't have any of them either because you're not having this RAG recombinase protein is non-existent in SCID. So that's a bit of foreshadowing for next month's Fireside Chat, where we'll have a professional from the Netherlands actually speaking on that more. That's something you don't want to miss, and I'll have more information on that later. And so now we have a functioning B cell receptor, and now we want to become a pre-B cell. So how does that happen? So we have this VCR on the top. Well, we have to show that it works. And the way we show that this B cell receptor works is that we expose it to antigen. If it's able to bind to antigen, it is selected to move on. And that is what we call positive selection. So here, this B cell was able to connect with an antigen here, the purple triangle. And so he's allowed to move on to the pre-B cell stage. But our friend here was unable to bind to anything. And so he's held at the pro-B cell stage. Really,
Austin, question here, is this still in the bone marrow?
Yeah, we are still in the bone marrow. That's a good question.
Okay.
And so, yeah, so now we have this selection process ensures that we're only using our energy on the B cells that have a working B cell receptor. So this is the part of encouraging foreign reactive B cells to move on and mature. So now we are in the pre-B cell stage, but I want to take a quick note because I'm showing a single B cell in all of these, but that is not the case of what is really happening. The pre-B cell stage in particular, we have a lot of proliferation. These cells are dividing into many different, well, really, I believe they're the same at this point, but we want to generate different clones of these B cells. And the reason is, again, to generate more diversity. There's another stage of recombination that occurs later on that I'm actually skipping over in this chat. But basically, once we have a functioning B cell receptor, we divide many, many times. And then those clones from the original B cell will then go through recombination again. And so that's how we get even more chances of hitting the right combination of DNA segments together, of those VDJ segments, to eventually create a functioning antibody. That's kind of skipping ahead a bit, but it's really important to note that this isn't just a single B cell going through the whole process. It is at the beginning, and then it diversifies. It really generates many divisions before moving on. And that was shown really well in Dr. Rawlings' slide again here, where there's a pro B cell and then multiple pre B cells. And these are held if they don't have BTK. And the reason this slide is important here is because BTK is part of the signaling process that tells these positively selected B cells to move on further in the maturation process. So we have a bunch of these pre-B cells with a working BCR now, and they move on to immature B cells. So the immature B cells then change the BCR into the first antibody. And these antibodies are actually bound to the cell surface at this point, and they're all IgM. So there are many classes of antibody, but IgM is the first that comes out. And we are still in the bone marrow here. And it's time for another round of selection. So in this case, we're in the bone marrow and we're showing self-antigen. And this B cell is very happy because it was able to bind again. And based off of positive selection, the one that binds to antigen gets to move on and mature. And this B cell is very worried because it's unable to bind to self-antigen. Well, it's a bit of a trap because this is a round of negative selection. So the B cell that actually binds to the self-antigen ends up being retained in the bone marrow and is basically told to die. It is not allowed to move on. And this is how we prevent autoimmune B cells from expanding and becoming mature B cells. If they are seen to bind to our self, any kind of antigen that is supposed to be in our body, they are told to stay in the bone marrow and not leave, pretty much. And so this B cell that was scared because it didn't bind to the self antigen actually gets to move on. And it does so by entering the blood and going into the periphery, such as the spleen, but it has to do something first. And so here we have these blood barrier cells basically asking the B cell for a password, and the B cell starts to think, what is it missing? It has an IgM. Well, it needs to do what is called class switching. And now we get another antibody on here, IgD, which is shown in the mature B cell stage on the bottom right here. So this B cell has two types of antibody on it, and that is the password to move on through into the spleen and the periphery. So we have now left the bone marrow and we've entered the mature B cell stage. Mature B cells live in what is called the periphery. This is anywhere besides the thymus, bone marrow. So there's evidence that B cells can live in tissues, like local tissues of infections. They definitely live in the spleen. And other than that's where they reside and are able to pump out antibody or do any of their other functions. They also have another ability to class switch. As I said, class switching is basically when you switch classes of antibodies. So our first immature B cell here went from IgM to IgM plus IgD. It can do many more types, such as IgM to IgG. There's also IgA, IgE, and that also helps to generate diversity. So there are many abilities of B cells. So as usual, they produce lots of antibody, excuse me. They're also able to present antigens. They're professional antigen presenters, And that just means they're able to show those pictures, like in vaccinations, to other cells and activate them. And they're also able to keep memory. And we'll see what that means. So here are some examples of other functions. The marginal zone B cell lives in the spleen and pumps out IgM. Plasma cells, they will exhaust themselves pumping out antibody during an infection. They're basically just there to use all the energy possible to produce antibody. And so the keeping memory part, I'm using a comic I stole off of Reddit as well. So here is a more immature B cell asking its elder, do you remember that last infection and how we kicked their butt? And the B cell does remember, and he remembers it well. And it's a bit traumatizing because whenever these B cells do fight infections, they end up causing a lot of damage to other things. The immune response is like a war. There is collateral damage all the time. Here you can see in the faded memories of this B cell, lots of his brethren unfortunately fell in battle. Lots of those plasma cells that exhaust themselves, pumping out antibody, die purely due to exhaustion, things like that. And then there's lots of damage done by other cells. That's a bit of a funny comic too. So in summary, though, this is an incredibly complex process. It was actually, if I were to do this again, I'd probably cut a bit of information out because there's a lot of steps, and I skipped over a lot of steps and only touched the surface on many, such as recombination. There are several steps of recombination, and I only showed the first one, really, for the B cell receptor. I didn't even go over how mature B cells get activated. That's another complex process involving other cells, including T cells telling B cells to turn on, things like that. And then it's also an incredibly inefficient process. So I showed you how these B cells proliferate, divide many times. And that's for a reason, to generate diversity, but also it can be seen as a bit of a waste. Nearly 90% of the B cells that are generated are told to die in negative selection. So it's a real use of energy, but that is all to protect from autoimmune cells being released into the body, released from the bone marrow into the body. And the important things to remember are positive selection, basically bind and move on. And then there's also negative selection for immature B cells, which is bind and die. And I find that to be really interesting in the B cell maturation process. This kind of double gatekeeping process to ensure that only the best B cells move on. Well, so why does this matter? Well, it's quite an amazing process. As far as I'm aware, B and T cells are the only human cells that actively alter their own DNA to generate diversity. And they're able to do so in a way that can create trillions of different combinations. Theoretically, any antigen, even something off of Earth, there might be a B cell that can react to it because of how much diversity we can generate. And it's also something not really appreciated. I didn't learn that our own human cells can mutate until college, really. And I think that's something that should be taught more often. And only a very few amount of cells will make the right combination of DNA to pass both positive and negative selection. Why does it matter for XLA patients? Well, we need to know what we're missing. And it's also very relevant for XLA gene therapy because the hematopoietic stem cell is the target. Any cell that is going to become a B cell has to go through this process. So if we want gene therapy to work, we have to understand how these stem cells that get delivered the gene therapy will eventually become effective B cells and help functionally cure XLA, hopefully sometime in the near future. And that brings me back to Dr. Rawlings' slide that BTK expressing B cells have a major selective advantage. So in positive selection, these XLA B cells without BTK, they may have a functioning BCR, B cell receptor, on their surface. But what needs to happen in order for these B cells to move on out of the bone marrow is there has to be a signal from the B cell, from the BCR, that it works. And BTK is the main protein that does this. Without BTK, you do not get the green light to go ahead. You may have a functioning BCR, but you will not pass go. Do not collect 200. You're going to stay in the bone marrow in the pre-B cell stage. Those with BTK can move on. So Dr. Rawlings made a really important point that they're using a BTK gene that is slightly less active than the wild type, than that naturally found in most people. And that is because if only a few of these stem cells get BTK, the few that do are going to be the only ones that move on the selection process. And really, that's why this is an important process to understand before moving on with gene therapy. We're entering a brand new world with gene therapy. I believe one of the first patients with Wiskott-Aldrich syndrome just got treated with gene therapy in the US. That's another immune deficiency. And it's time for XLA to start pretty soon, which is really exciting, but we need to understand how this will all come together. And so that's really the end of my talk on B cells. And it's time for questions. How did you like the comics? Yeah, Keith, if you have any fact checks.
I think that was outstanding. You've put in tremendous effort for this, because I think it's one of the most complex processes too, in that there are pathways we don't understand with B cells that we understand with T cells. I mean, we do know that T cells go to school in the thymus, and I am always intrigued, and I don't know from your end, because you probably are more abreast with the literature here. How does negative selection come about in the B cell, and how does is that different from T cells? What do we know? Because we know in T cells there's this gene called AIR that makes sure that any type of protein in the body is expressed in the thymus, so that those cells can recognize whether it's an insulin producing cell, or whether it's a testicular cell, or whether it's another gland or muscle that can be seen in the thymus. What do we know about that in the bone marrow?
I was struggling to find answers to that exact question, because I was asking one of my friends who's in the med school here, what is the signal for negative selection? Like what tells them to basically die when they bind to self-antigen? And you're right about AIR as well. What in the bone marrow would express like insulin, for example? I'm not sure that there is. So I actually didn't find an answer myself for that question. But I had it in my mind, and it was a little frustrating because I couldn't find it. And I was like, I'm missing a huge, important answer here. And I guess we're just missing it entirely in terms of science.
And as you point out, with gene therapy, the immune system has like 20 times more brakes than accelerators, so to speak. And it's like a martial artist that has all that power, but knows not to use it. And we saw with COVID, when it goes crazy, it can really go crazy. And people die from excess inflammation, not from actual virus itself. And there's always been that concern with a lot of gene therapy, are we going to get good cells, or will they, those cells become, sometimes they're concerned for malignancy. for example. And one of my thoughts is that, is there anything that happens, or does an aging body affect negative selection as opposed to a baby, that we don't yet know to kind of tame those, highly proliferative cells that could escape?
Yeah, another question that I really struggled to find an answer with. I was amazed when I was going through my notes back from school, and all of the positive selection, negative selection notes I had were all on T cells. It was hard to find stuff on B cells, believe it or not. Now I remember I did have notes on, so if I go back a little bit to, where was it? Here, basically, it's not entirely, I made a very, I painted it with a broad stroke here. I ignored receptor editing, like if you bind with mild affinity to self-antigen, there's a chance to go back and try another combination of, are we at the heavier light chain here? I forget. To see if it'll not bind to self-antigen. And also I ignored anergy, where, okay, you bound to self-antigen, we're still going to let you go to the periphery, but you're not allowed to activate. Things like that I skipped over, but I struggled to find anything more specific on negative selection. I really wish there was more out there. It took me a while to even find the stat that 90% of B cells generated will die in negative selection as well.
And as I recall, just recently in fellowship, I remember Janeway saying, we don't know much about this, and I was wondering, I don't think there's been any new information, but
I couldn't find any. Yeah,
this is really fascinating.
Yeah, what do you think of the comics? I'd love to see these in scientific publications someday.
I think they're great, because you can tell you're
They're
passionate
helpful.
Yeah.
I was just
Very
going to say,
helpful.
what I think they'd be really helpful in educating people newly diagnosed with XLA, right?
Yeah.
And
Yeah,
how to really
yeah.
explain it to them,
Yes.
right?
Yeah, no, I had a really fun time drawing these. It was a lot of work, but I thought it was worth it. And as
It
soon
brings,
as
it brings this really technical scientific
Right.
information down to the layperson,
Yeah.
when you do it like this, makes it kind of understandable.
Visuals are always good like this, right?
Yes. Yes.
So you said you referenced what Dr. Rawlings said about, and I remember this presentation where and I'm not sure if I understood it correctly, but he said something about you don't have to, maybe with the gene therapy, you don't have to generate, a large number of, successful B cells. There was something about you, if you just get to, say a certain critical mass, that's good enough. I mean, it's effective enough. Was that your point about how this selection and process works, or something like that? Or
yeah,
that?
very much so. So even if you, let's say 10% of hematopoietic stem cells take up the BTK gene therapy, those 10% will be incredibly more likely to pass through positive selection than those that don't. So the thinking is the body will see that a few of these cells are able to get the go-ahead signal, and that will make up for all the cells that didn't take up BTK. The cells that did will go through and proliferate enough that we get enough diversity from those few cells that did. Now, Dr. Rawlings would be able to speak on that much better than I, but I believe that's the line of thinking. the selective advantage is basically the reason there are no B cells in the periphery for XLA patients is because of this stop. The stop here. You need BTK to, it's basically the driver's license to move out of the bone marrow. And so any that do will be far more likely to pass positive selection. And hopefully that makes up for it.
Yeah, and he said that in his mice experiments, it showed that even though it was a relatively small percentage, seemed to be almost as efficacious as a normal mouse, right? Like it was approaching like 80 to 90% of the effectiveness of it.
Yeah, I want to say the 80% number.
Yeah, something like that.
Cool. Well, thank you for joining, everyone. Anything else?
Well, you teased us about next month's Fireside Chat.
Yeah. So next XLA Life Fireside Chat will be August 23rd, time to be determined. We have guest speakers from the Stop XLA program in the Netherlands. So they are trying to provide evidence that newborn screening panels for XLA is cost effective and will improve quality of life. So they are looking for XLA patients to join. They're trying to get more physicians involved as well. As I said, they're from the Netherlands, so we have their Dutch logo here next to the English one. And I'm really excited. They'll be our first international guest speaker. And they're really important program, I believe, and Jeffrey Modell Foundation is very supportive of their project as well. So we're hoping to create a mutually beneficial project, and we'll introduce the Stop XLA program to the XLA community next month. Late next month.
Hey, Austin, sorry.
No, go ahead, Andrea.
Are all the firesides going to be on your website?
Yeah, so they are up on our YouTube page, and I've posted them in the blog section of the website.
Okay.
Yeah,
Well
I'm
done.
excited to go international next month.
You've got an abstract accepted at the European Society of Immunodeficiency. So
Yeah,
congratulations.
it's possible we'll be in Sweden in October. We'll see about that.
Well, that's great. Then you might meet your Netherlands colleagues there, possibly.
Yeah, that would be amazing.
Oh, that's cool. Very cool.
Yeah, I'm very excited for this project. It seems like a no-brainer to put it on the newborn screening panel, but people, whoever the powers may be, are demanding a lot of evidence and a lot of they want a monetary cost, like
It's insurance carriers, I'm sure.
Yeah.
A cost benefit analysis.
Yeah, exactly.
Pretty much. And so I'm hoping, it seems like a no-brainer, and I think the XLA community will be excited to join and help out. So
How prevalent is XLA in the general population?
1 in every 200,000 live male births.
Oh, okay.
Or was it live births total?
Now, severe combined immune deficiency before screening was thought to be even up to 1 in 300,000 live births. And now the prevalence has increased with screening in the US to 1 in 50,000.
Whoa.
I can attest to a kid from Malta who had recurrent meningitis, who I knew of when I was away, and was actually unfortunately passed as an infant and was diagnosed with XLA after the fact. So there are cases out there, and like this is one case I know of in Europe. Yeah.
Wow.
I forget where I heard, but the prevalence of like CVID, and I think I don't know where I heard it, but the prevalence of XLA is increasing, and it's gotta just because of better diagnostics. And so if we get it in newborn screening, that's a huge jump from 1 in 300,000 to 50,000.
Yeah.
Mm-hmm. Totally. And, yes, the VID was 1 in 50,000. Now we say it's 1 in 20, 1 in 25,000. I mean, if you go to countries like Kuwait, it's 1 in 10 to 1 in 15,000.
Wow.
So yeah, I'm here in the Southwest, and I can tell you, in terms of immune deficiency, I have my 4th kid going into a transplant evaluation. I mean, there's over 450 immune deficiencies, but there's a lot of need for sure, and it's very under-recognized as a unit.
Mm.
So, yeah, so it's more, these diseases may not be quite as rare as people think, just that
Right.
they're underdiagnosed.
Mhm.
Yes.
Right, and the cost benefit is if you don't diagnose it early, how much do you spend on it? That's the case that's got to be made to the insurance carriers, right?
Well, how many hospital visits is it worth?
Right, right,
It's,
Yeah. Meningitis, pneumonias,
right,
infections.
right.
But the positive side could be then if it's more prevalent than people have so far thought, then the cost benefit of developing a genetic therapy cure is that much improves that business case,
That's right.
so that you eliminate
Good
it.
point. Yeah, exciting times. I mean, this is a real turning point in immune deficiencies we're seeing right now. Like, as I said, the first Wiskott-Aldrich syndrome patient, I believe, just got treated for gene therapy in the US very recently.
What medical center was that at? Do you know?
Can't remember. Not off the top of my head.
So I think that the graphics are very promising, and if like some of those could lend themselves to animation too, that would be really
Yes.
cool.
Oh, the comics.
Yeah, yeah.
Yeah, I'd love to. I actually kind of like how raw they look.
Yeah, no, right.
I can actually, let's see, can I bring up my Microsoft Paint? Here we go.
Yeah.
Here's my whole worksheet.
Like the Ricky and Morty of disease.
Yeah. No, I mean, I would love to keep drawing these in so many different ones.
That's really good. No, I think
Very good.
promising.
Yeah.
Yep.
And it could apply to more than just B cells, I
XLA.
think.
Yeah.
That's right.
No more bio render, Keith. I'm gonna hand draw my own from now on.
Pretty cool. It's awesome.
Excellent fireside chat, sweetheart.
Awesome.
Good.
This was outstanding. Yep, I really enjoyed it.
Awesome. Well, thank you everyone for joining. And we'll see you next month. Okay, I'm gonna start
See you next
the
month.
recording now.
Thank you.
Bye.
Yeah.
Bye.
Bye bye.